Good afternoon, everybody. Today, it is a pleasure to me to introduce Professor Adil Mardinoglu. He is Professor of Systems Biology at the Center for Host-Microbiome Interactions at the King's College from London. And he is also group leader at the Science for Life laboratory of the Royal Institute of Technology of Sweden. Dr. Mardinoglu is working in system biology, and he is studying, for example, how biological networks can be used, for example, to study the reprogramming of liver physiology in response to non-alcoholic steatohepatitis or non-alcoholic fatty liver disease. So it is a pleasure to me to present him and to introduce him to you. It is time to start. So thank you everyone to be here, and thank you to Professor Mardinoglu.
Perfect, thank you so much. Again, thank you so much for a very kind invitation and very nice introduction. So, but may I ask you before I start, what is the background of the people who are joining here?
Well, we are molecular biologists. We are also doing some in silico analysis, especially me and some colleagues using omics dataset, but mainly we are molecular biologists.
Excellent, excellent. Perfect. So in that sense, I can start. Again, thank you so much for a very kind invitation. And we are basically, I work both in King's College London and also at SciLifeLab Laboratory in Stockholm in Sweden. The main things, we have groups in 2 different places, and we are mostly working on systems biology and systems medicine applications in detailed characterization of the patients. And then later we find some targets, and the moment that we think we actually find something, we initiate some startup companies, and then we start developing actual drugs for the specific target that we identified. But before we get into the details of the project, I want to briefly tell you about systems biology and systems medicine. I'm sure you heard of all this terminology, but please let me go through it from our perspective. The thing is, I will not get through its details, but we are aware that we are dealing with a huge complexity. Like, we are dealing with the human body, and over the years, the complications are actually, the complicated circuits are building on top of each other. And then considering the effect of the diet, genetic differences, and then the environmental effect, it's actually, the problem is getting more difficult to solve, like especially at health and disease stage. So our methodology is, we are really, instead of focusing on individual component, we are really interested in focusing the entire complex system. So if you look at the Rube Goldberg invention, if you really want to understand the system, you really need to know first how many components are there, what is the dynamic or characteristic of each component, how they are connected to each other. Then actually you can have a global understanding of the system dynamic. And that's exactly what we're trying to do in the human body as well. Please consider one thing. In the Rube Goldberg invention, if you really focus on one component, like let's focus on time, all right, like clock. So you would mostly think about the things about, time and associated things, all right? But when you look at it in the system, it's actually, it has nothing to do with the time. It is simply, he's simply using it to cool down his soup, and it is kind of independent than the time. So that is quite important for us. So our methodology is, we are really interested in the generation of the genome-scale metabolic models, or in other terms, biological networks for individual cell types and tissues. And then later we actually plug them together, and we can study the interaction between different components, and eventually we can plug them together and run the whole body simulations in health and disease state. I'll give you some examples of it, but let's look into what is these models, like what do they do? Genome-scale metabolic models are simply, from the name, you can see that it's actually the representative of the metabolism that is present in a specific cell and tissue. So we collect all the biochemical reactions informations, and this is actually, look at here, it's conversion of glucose to glucose 6-phosphate, and it's at the end, it's actually a coded, like associated to one protein coding gene, which is the GC key. So basically, we have the metabolite, we have the reaction, we have the metabolites, we have the reaction, and we have the gene information, all right? So we gather all this biochemical reaction information in the human body first, which is actually more than 10,000 biochemical reaction now. And then if you look at the individual cells and tissues, it is something between 5,000 to 8,000, which are considering the differences between different cell types and tissues. So the moment that you gather this biological network, you actually generate this stoichiometry matrix, which is in the center. Simply reaction 1 is presented like glucose consumed with -1, glucose 6-phosphate with 1, and then you see ATP is consumed and ADP is produced. So basically, you gather all this biochemical reaction information into the computer, and the moment that you have this network topology in your computer, then you can perform network-dependent analysis and model simulations. Let's look at it in another way. We are simply making the Google map of individual cells and tissues. And the moment that we have the Google map, you can actually use it like we are doing in the Google map algorithm. We can actually go from one location to other location using the shortest path, fastest path, or any other alternative paths. And that's what exactly we do. And then the moment that you have generated these models, you can actually integrate different omics data, including proteomics, metabolomics, because metabolomics, proteomics, and then transcriptomics, and fluxomics. Simply, we have the metabolites, please remember, and the metabolites are, forming the reactions, and that is catalyzed by specific genes, which is like protein coding genes, which you can actually directly integrate proteomics and transcriptomics data. So once you have these models, you can actually study the different molecular mechanisms in a health and disease state, simply by revealing the differences on the based on proteomics, metabolomics, and transcriptomics data. Of course, if you have these models, you can run whole body simulations and you can study the interaction between different components. And please don't forget, we always look for a new drug target or new markers that could be used actually in personalized and translational medicine. To date, we have generated adipose tissue model for studying the adipose tissue and obesity and also diabetes. We've generated the liver tissue model for studying the fatty liver disease, liver cancer, HCV related indications, and muscle tissue model for type 2 diabetes. And with the recent algorithms that we developed, we have actually generated models for more than 80 different cell types and more than 50 different human tissues based on transcriptomics and proteomics data. If I give you a brief overview of the data that we have used in our analysis, we have simply generated extensive amount of proteomics data in the Human Protein Atlas project, and we studied the concordance of the proteomics and transcriptomics tissue using the data generated in the HPA platform and also in the Phantom and also GTEx consortiums. So this is actually a great resource for studying the molecular repertoires of individual cells and tissues in the human body. We have done the same thing for the cancers as well. We simply we simply integrated, performed the systems level analysis in the TCGA data and also with our own data that is generated in-house. And eventually we actually identified the prognostic clusters and the driver genes involved in the progression of the tumor and associated them, studied the associations between the clinical survival of these patients. So we in addition to the proteomics and transcriptomics data, we also generated extensive portals for studying the human gut microbiome atlas in the human gut microbiome, and we simply studied the variations between the different metagenomics data in different disease, considering the effect of the genetic factors from different countries and different continents. So we simply studied the functional composition and association of these individual species and the bacteria for the development of specific diseases. So with that, I want to tell you some examples of how we use all this information in the treatment of fatty liver disease. I will simply present you two different projects here, in which in first where we identified a target and we start developing a drug for that. And also, I will give you a detailed example of how we actually identify the new mechanism and then use it for development of the patients, not only for fatty liver disease, but also other metabolism-related diseases. So please let me tell you a standard, a drug development project, which is actually a target that we identified 3 years ago, 4 years ago, almost now. And then now we have developed thousands of molecules for that. And eventually we identified a list of targets, which can be actually which is now in tox studies, and we are hoping to initiate the phase 1 study within the 2022 or latest the beginning of 2023. So in this project, we simply identify a novel target, and then we perform a new drug development project. And in parallel to that, now we tested the efficacy of these compounds in the animals. And after seeing the fantastic effect that, that we deserve actually after a huge amount of work, and now we start generating this under GMP conditions, and we have performed GLP tox studies. And then with that, we simply, with that, we simply started all this information by generating a liver model, genome-scale metabolic model for hepatocytes in the liver tissue. And with that, we use actually transcriptomics data, and we identified new targets that can be used for the treatment of the patient. So if I tell you briefly about the generation of the models, we simply integrated all the biochemical reaction information in the literature in a large database. And then we generate transcriptomics data for NAFLD and for healthy. And eventually we integrated with transcriptomics and proteomics data. And we actually made a model, comprehensive liver tissue model. And we use this model for the analysis of the transcriptomics data in the fatty liver disease patient, and generated for fatty liver disease patient. And later we identified some mechanistic differences. If you look at the differences, this is actually the snapshot of the computer output. We simply map the transcriptomics data into our network from healthy and disease. And in this case, the blue ones indicate the downregulated reactions, and the red ones indicate the upregulated reactions. Here, one thing interesting we found is there's an actually consistent and significant downregulated around serine metabolism and around glutamate metabolism. And of course, this is all about the, the screenshot, the snapshot of the metabolism in patients with fatty liver. When we further investigated the differences, like what is the potential reason for such deficiency, we actually found that there is a decreased downregulation, a decreased synthesis of the serine in this patient group. So basically, there is a deficiency, and this is due to the decreased synthesis, because this is normally an amino acid that can be synthesized in the human body, in the liver tissue, and this can be actually converted to glycine and eventually can be used in the human body. So the main thing is, at the time, this study has been published in 2014, but we didn't know actually much more than that in terms of mechanism. We presented as the deficiency around serine metabolism. But we later designed another study where we actually had 86 different subjects with, with the increased cardiovascular risk, with the high liver fat, and then we actually generated VLDL kinetics data in this patient group. We also collected extensive amount of plasma samples for generating of metabolomics data and lipidomics data, and we integrated all this data together with the VLDL kinetics using a holistic approach. In this case, we actually integrated the liver tissue model with the adipose, muscle, and brain tissue models, and we included the effect of the red blood cells, and eventually we actually studied the in and out to the liver tissue and predicted the intracellular metabolism of liver tissue. Considering that we measured the VLDL kinetic, considering that we measured the adipose and fat mass, adipose and muscle mass, fat mass and muscle mass, and we actually calculated the glycerol, fatty acid, and ketone bodies, and also amino acid secretion. So eventually we basically integrated all these models and predicted the intracellular fluxes in the liver tissue. One thing which is interesting is, after running all this comprehensive analysis, we found that there is an actually a significant association between the reactions catalyzed by NNNT and the GSR with the independent immediate liver fat, which is actually a very good indication for predicting the risk factors for fatty liver disease and for NASH, and also for cardiovascular diseases. And when we further investigated, we found that these two reactions are actually mostly involved in the synthesis of the NAD and also antioxidants. Simply, our analysis showed that the deficiency, the amount of the liver fat is significantly associated with the NAD and the GSH levels. When we further investigated, we actually found that this is mostly due to the deficiency around glycine and serine metabolism, which actually allowed us to link it to our earlier study. As I mentioned, we also collected plasma samples from these patients, and when we measured that, we found that there is a serine, glycine, betaine, and acetylglycine deficiency, which is actually the compounds that can be converted to the serine and glycine in patients with fatty liver disease. As a result, we found that there is a deficiency in around serine and glycine metabolism from our 2014 study, And in 2017, we were able to actually, link it to the deficiency in serine. 1 second, please. I want 1 minute break. And then we also integrated to the metabolomics data, and we actually found that the list of the metabolites are significantly downregulated in this patient group. And this is again mostly linked to the serine, glycine, betaine, N-acetylglycine, and also the other key metabolites that are heavily involved in the progression of the fatty liver disease, including curinate, carinate, and also other metabolites. So we also, after seeing the effect of the serine and also other metabolites, we wanted to see if we can actually modulate the serine metabolism by simply supplementing this metabolite, because it's a natural amino acid. It is actually included in the diet. We said, what happens if we actually provide this as a supplementation to the people? Because we really wanted to see if we can actually modulate this serine metabolism by simply administering it. And then we designed a proof of concept study with 6 subjects. And then after 2 weeks of serine supplementation, we simply found that the amount of the serine in the plasma is increasing, and also the liver tissue function, including ALT, AST, and all the clinical parameters are significantly decreased, which is actually a very big surprise for us, that even the liver fat of the patients are significantly decreased within 2 weeks of serine supplementation. Again, this wasn't a conclusive study. This was just a proof of concept study to see if we can actually modulate the serine metabolism and affect the metabolic parameters of the patients with fatty liver disease. But our actual analysis told us much more than that. Simply based on our analysis, the moment that we can actually modulate the oxidation of the fat by promoting the uptake of the fat, which is L-carnitine tartrate, and then promote the oxidation of fat with nicotinamide riboside, nicotinamide mononucleotide, or niacinamide, and eventually linking to the serine and NAC supplementation to boost the formation of antioxidants, we can actually activate this pathway, activate the metabolism, and remove the excess amount of the fat in the liver. That was the main finding of our study. The moment that we had such a hypothesis, we start actually we gather all these compounds, and then we start running these animal studies, and we tested the efficacy in mice. And it was very interesting to show that in 2 weeks, we simply decreased the amount of the fat on more than 50% in the animals, which was a very good result for us, to be honest. And then we also performed the same experiments in rats, and we showed that the efficacy is super good compared to individual metabolic activators and compared to control. And also we seen these changes in the liver fat level, and also in the clinical markers that are heavily used in the diagnosis of the fatty liver disease. In addition to that, I want to give you one more example, because the main thing is our hypothesis, liver tissue has a capacity to burn the excess amount of the fat if you have enough or required materials to do that. So I want to give you now one more example to actually prove this concept. In this context, we have actually designed an isocaloric carbohydrate restricted diet in subjects with fatty liver disease. Simply, we had 10 patients with fatty liver disease. They have, as you see from the parameter, they have high liver fat in their liver tissue. And the main thing is we wanted to see, before they start to study, we first calculated their calorie intake. And then finally, we changed the carbohydrate level from the subject's, carbohydrate intake of the subject with more fat and more protein. Please remember, they have more fat in the liver, more fat in the plasma. And eventually we provide them more fat and more protein, and we wanted to see what happens if there is no new fat synthesis. So simply we are here measuring, we are testing the capacity of the liver tissue, if it has a capacity to burn out the excess amount of the fat, if there is no new one, if there is not de novo lipogenesis. In this context, we generated this multiomics data. Simply they come to the hospital every day during these 14 days of study. And then we generate extensive amount of phenomics data, including de novo lipogenesis, and then lipid parameters, anthropometric parameters. We collected liver tissue biopsy from transcriptomics. We collected, plasma samples for metabolomics and inflammatory biomarkers. And we collected fecal samples for metagenomic samples at day 0, 3, 7, and 14. And then we start studying the functionality in the microbiome, and it's you see the significant shift at day 0 and day 1, which is actually the changing of the diet, and day 3 and day 7. After 7 days, it is actually saturated. We also look at the functionality by simply measuring the short chain fatty acids and by generating metagenomics data. And again, you see that there is a significant decreases in the short chain fatty acid level. When we studied the functional differences based on metagenomics data, with the shotgun metagenomics data, we have actually seen that there is a consistent and significant changes around starch and sucrose metabolism, here you see, and also around folate metabolism. And when we further investigated, actually, of course, starch and sucrose metabolism was an expected outcome because of the changes in the carbohydrate intake, but folate biosynthesis was a very interesting surprise for us. When we further investigated, we have actually found that there is a significant decreases around folate biosynthesis in the bacteria in the human gut. And we, in addition to that, we measured the serum folate levels. We wanted to see if these changes in the gut is actually reflected to the human plasma or serum. And in that context, it was very interesting to show that we have actually, there is a significant increase in the folate level. Of course, after seeing the differences in the folate, we wanted to study the folate metabolism in the liver, and we look at the transcriptomics data, and here you see that there's significant differences in the folate metabolism based on transcriptomics data of the liver tissue. And we further integrated this data with the liver tissue model that we generate. And again, here you see there's a significant decrease in the glucose intake as expected, and there is a significant increase in the fatty acid oxidation. And very nice surprise about the increased branched-chain amino acid catabolism. We know that branched-chain amino acids are associated with the insulin resistance and the metabolic alterations. And again, it was very nice to see that there is a significant decrease in that. And one very good surprise for us was the activation around the methionine folate cycle, and actually very much linked to the serine and antioxidant metabolism. So this is clearly showing that liver tissue has a capacity to burn the excess amount of the fat if there is no new fat, and then to decrease the amount of the fat in the liver. And this is very much linked to antioxidant metabolism, NAD metabolism, and serine and glycine are actually have a key role in the in this process. So the moment that we have these proof of concept studies, as I mentioned, we run this serine research study with 6 subjects, and then we actually produce this formulation under GMP condition. We run a phase 1 study. Since even though these are natural substances, considering that you are using high doses, considering that you are using actually, making a new combination, you need to run a phase 1 study. And then later we start running phase 2 studies in NAFLD, in Alzheimer and Parkinson patients, and also in COVID-19. I will also tell you why we run such studies. I will tell you the background of that. So let me tell you about the phase 1 study. Simply, we wanted to see what happens to the patients the moment that we provide the substances. And in this context, we have actually gathered 10 healthy subjects. We put them in the same hotel, and then we give them the same breakfast, and then the dinner, same dinner before the day, before the start of the study, and the same breakfast at the same day. And we collected blood at T0 and provided supplement, and then we collected samples during the day. And then we brought the same subjects to the hospital again after a washout period. And then in there, we give actually 1 glass of water instead of formulation. And here you see the kinetic of serine and other metabolites. And it was very interesting to show that the amount of the metabolites that we supplemented are significantly increased in the plasma. And then this actually affected a huge amount of metabolic parameters that are significantly associated with fatty liver disease. And also, before we start the study, in the phase 2 study for fatty liver disease patients, we wanted to generate a multiomics data for a group of subjects, and we actually identified the clinical markers and key parameters involved in the progression of the fatty liver disease based on clinical and physical variables, proteomics, metabolomics, and oral and gut metagenomics data. Here you see the output of the analysis, and you see the key parameters, key clinical parameters involved in the fatty liver disease. Inflammatory protein markers, you see plasma metabolomics markers, you see, and then oral and gut metagenomics data. Basically, this is the baseline data. And in this cohort, we also selected number of these patients, and then provided the formulation and see what happens after a certain period. And I will also tell you about that. So based on these parameters, we predicted the liver fat level based on omics data. And here you see that the metabolized clinical parameters and also oral and gut metagenomic parameters are quite important for prediction of the liver fat. And here you see the predictive value. And we have, to be honest, we have found a very good markers for prediction of the liver fat. And the moment that we identify, we characterize these baseline subjects, we also wanted to provide the formulation and see what happens in this patient group after certain time. And in that patient group, we again we provided the formulation, combined metabolic activator formulation in the placebo controlled study. And again, we screened this patient, like we provided supplementation for 70 days, and we generated the same data, including MRPDF liver fat data, metabolomics data, plasma proteomics data, oral and gut metagenomics data. And we also phenotyped these patients genetically and then studied the genetic variations. And eventually we found that the liver fat, ALT, AST, and uric acid levels are significantly decreased only in the active arm compared to placebo arm. And here you see the key parameters affected based on oral and gut metagenomics data. And then here you see the effect of the oral and gut metagenome on the plasma metabolites, which is actually very interesting to see the association between oral and gut microbiome and the plasma metabolome. And eventually we reported the differences on the uric acid level, because it was actually a good surprise for us to see this significant decrease in the urea levels. And we also found that there is a significant association between liver fat, uric acid, and supplemented materials. And one clinical parameter was FGF21 based on proteomics data, which is actually quite interesting for studying the association between the combined metabolic activators, clinical parameters, and the liver fat. And we also studied the variation between the oral and gut microbiome in the clinical study that we performed. And it was very interesting to see that the bacteria that have been significantly associated with the liver fat has been significantly and possibly affected by the supplementation of the combined metabolic activators. So here you see a summary result. Basically, with the supplementation, liver fat, ALT, AST, uric acid, and creatine level are significantly decreased. We found that fat test oxidation is significantly increased in addition to the decrease in the urea cycle, curinine, curinate, and TMAA. And also we found that inflammatory protein biomarkers are significantly decreased. And also we found that the abundance of oral microbiome and gut microbiome are significantly affected with that formulation, supplementation of the formulation. So the main thing is, after seeing the significant improvement in the metabolic activators in the metabolism, we wanted to test this formulation in the COVID-19 subjects, because the main thing is, at the time I was actually involved in the management of the COVID-19 patients in Turkey as a representative of Turkish Ministry of Health, and we basically seen significant effect of the COVID-19 on the liver tissue functions. So basically, liver tissue functions got worsened, and then the amount of the fat in the liver is increased and affected, and also affected the ALT and AST level. So we simply wanted to see if we can actually provide this supplementation to the patients and see if we can actually help these patients in improving their metabolic parameters. So in that context, we started phase 2 study, an open label phase 2 study. And with this patient group, it was actually quite interesting because we wanted to see what happens with the clinical parameters, but at the same time, we start recording the symptoms of the patients. And this was an open label phase 2 study. And after 2 weeks of supplementation in around 93 patients, we found that the ALT and AST parameters are significantly improved as we expected. Here you see the differences. But more interestingly, the actual recovery of the patients are significantly improved as well. So after seeing such positive results, we wanted to see if we can actually test this in a double-blind placebo-controlled study. And again, out of 300, in testing of the supplementation in 304 patients, we found that the supplementation administration of the combined metabolic activators accelerated the recovery of the patients more than 3 days compared to placebo group. And one of the good news is actually in open label phase 2 and phase 3 studies, no one has been hospitalized in our study. And then in the active arm, also a number of patients are hospitalized in the placebo arm. So here you see the next generation plasma protein profiling. So we also studied these variations in the plasma protein markers in COVID-19 patients with and without formulation. And it was very interesting to show the significant differences and the improvements with the formulation of the combined metabolic activators. As I mentioned, we generate extensive amount of multiomics data for fatty liver disease, and we have also did the same thing now for Alzheimer and Parkinson's disease patients, COVID-19 patients, for healthy, for obesity, and eventually we gathered all this data in an interactive and visualization and database of multiomics data at iNet models. And now you can simply get to the portal and you can study the variation between different clinical parameters and with different omics data. And with that, I would like to acknowledge my collaborators at SciLifeLab at University of Gothenburg, Helsinki University, Imperial College London, Istanbul Medipol University, University of Groningen, and King's College London. And also we have very good collaborators at Stanford University and also at NIH and also in University of Leipzig. Of course, such study can only be done with extensive amount of funding. In that context, I would be very happy. I mean, I want to acknowledge Knut Alice Wallenberg Foundation, Chan Zuckerberg Initiative, and Bill and Melinda Gates Foundation for their extremely generous funding. With that, I would be happy to answer your immediate questions if there is any. Thank you so much for your attention.
Okay, thank you very much, Professor Mardinoglu. So if somebody want to do some question, please, this is the time. So.
Pablo, I have a question.
Yes,
well,
me too.
Can you remove your slides so we can see each other?
Of course, yes.
Okay. Hi. Now I have a question. I mean, I think that your data are very convincing and very stimulating. It's very complicated. I saw that you ran several studies. Did you ever consider gender difference?
Yes. Yes. The thing is, very nice question. The main thing is, like, for now, we actually tested the formulation in more than 1,000 subjects. All right. And now what we are doing is we are actually performing a very detailed analysis on the gender, based on gender. But the thing is, as you would agree, based on phase 2 studies for NAFLD and Alzheimer and Parkinson's disease, the number of the patients were not really convincing. But with the COVID-19 patients, we have tested this more than 700 patients. In the short glimpse, I can tell you that we didn't really reveal much differences, but now we are in terms of clinical outcome, but now we are performing the sex differences, gender differences in the multiomics data. Because that is something, in that study, we are actually expecting some differences, especially in the protein markers and also in the metabolite level. And we will inform you about that. I mean, we can inform you.
I think that this is crucial for 2 reasons. There are strong data telling that the outcome is different according to the gender. And second, also the biomarkers are different according to the gender.
I fully agree with you.
I'll give you one example. Uric acid, for instance, is different between males and females. So I wonder whether this is, there is something, we work on that, and we should actually plan studies by dividing patients or subjects according to the gender. And I think that your data can really help us to open this black box. And I mean, the gender issue has been largely disregarded in studies.
But to be honest, with the uric acid, I actually agree with you. Even though we had a very small number of subjects, we actually see a very clear decrease in the male subjects. In the female, that is always noisy.
Exactly.
I fully agree with you. So, but what we can do is, what we are doing is now, we are simply we are testing it in the COVID cohort, because in the COVID cohort, we have now 700 subjects in total. And with that, we can actually perform such analysis. But to be honest, we didn't want to provide a solid result based on phase 2 studies because the number of the subjects were too low and it would be speculative. So, but in COVID-19 subjects, we can do this. We are doing it actually.
Okay.
Very good question. Very good comment. Thank you.
Thank you.
Also, I was thinking to do the same question, so
Sorry, Pablo.
No problem.
I've stolen your question.
It's okay. So, somebody else had a question for Professor Mardinoglu?
So basically, Pablo, our methodology is we are really interested in generation of this multiomics data. First, detailed characterization of the subjects. Once we characterize the patients, we always come up with new targets because we are always looking at the mechanistic differences. But the main thing is how good it is. We need to perform animal studies. The moment that you start a new mechanism or new drug, then we want to run clinical studies. But again, during the clinical study, we want to run again this multiomics data, since we can actually see which parameters we really modulating. Because at the end of the day, I mean, in the fatty liver disease study, we wanted to really run the study as a whole because we wanted to characterize at the beginning and then see what parameters are changing, what else we should do to further improve it. So that's why even clinical studies are generating new questions and new answers, which is actually making it more interesting based on multiomics data. And the challenge is to integrate and to analyze this data. And again, the systems biology, systems medicine, and now AI actually is helping us a lot in integrating and in understanding this data set.
Yes, one of the things that I find very difficult is that the data that we manage is always looking a picture in patients, only at one. We are stratifying the patient, for example, according to the histology in the liver, but we cannot follow the patients on time. So for this, the Mooring models are ideal to integrate to obtain better models about to integrate these annotations about transcriptomics and proteomics and metabolomics. Because if not, we are always looking only we don't have the film, we have only a picture of the situation.
Pablo, I want to show you actually one slide. I mean, this is, you said mouse studies, but now with the volunteers, the things are getting very easy as well. Please look at this study, all right? In this study, we actually recruit 10 patients, and these 10 subjects are really guys who believe in the science, all right? And the main thing, we were able to convince them and get 2 liver biopsies within a week. I mean, you know that this is technically impossible. You cannot even do this in the animals, all right? But here, even the patients volunteered for it, which was a very nice study for us. And this is actually, such studies should be performed in larger cohorts in different diseases, because with this study, we simply had chance to follow up the patients. We had chance to record the diet because we provide the diet to these people. We prepared and give them. So we knew basically what we gave, all right? And then by generating this data, this actually allow us to understand what is happening in fatty liver disease, which paths are activated, how things are evolving. So I fully agree with you that we need really good animal studies. But of course, by generating such detailed human studies, we will also have a lot of answers from such studies.
Thank you to all to be here, and thank you especially to Professor Mardinoglu for all the information and the inputs that you give to us.
Pablo, again, thank you so much for your very kind invitation. It was my pleasure to be part of this program. And the main thing is that if there is any collaboration opportunities, please drop a line. We are very much interested in working with others. We have a very big team, like we are more than 50 people in my team. So we are always open for nice, good collaborative projects. And we would love to collaborate if there is an opportunity, basically.
Well, thank you very much. This is very promising.
And I also want to give you a little bit, I mean, like about the details of omics data generation. To be honest, like multiomics data is something very fancy, but it is expensive. But we also work on decreasing the cost of it. So basically, we open our facilities in Istanbul. We have a facility in Sweden, so where we can actually generate all this data with super decreased cost. So if there is any need, if you need any help on generating any of these data sets, please get in touch with me. I mean, we will simply generate the data with in a non-profit organization, and we can support that. We can support your projects as well. As I mentioned, like we are generating huge amount of data. Eventually, we made a special agreement with Illumina, and we had our own facility, and we only pay for cost basically. Just to let you know, all right?
Okay, thank you very much. That's a good thing. That's a good thing to hear from you. Okay, have a nice day, and thank you
so much.
again.
Thank you. Bye-bye.
Bye. Thank you.
Yeah, bye.
Bye-bye.
Bye.
Bye.